these guys aren't, right? Because to be a sister chromatid, you have to be genetically identical. And these guys are not going to be genetically identical. So anyways, in prophase 1, what's going to happen is these non-sister chromatids are going to exchange pieces of DNA in a process called crossing over. So literally, these non-sister chromatids, they cross over one another, and there's a mechanism in your cells that allows you to kind of cut and paste DNA. That's pretty much what we're doing. We're cutting and pasting this portion, and we're just switching, basically switching sides. So this guy will still have here, and then this guy will be like that. So you're making these homologous chromosomes, you're increasing the genetic diversity. So instead of just having this and this, which are exactly the same, now we have 4 unique pieces of DNA. And so in meiosis 1, again, after this crossing over occurs, we're going to have these homologous chromosomes are going to be separated from one another. And then in meiosis 2, that's when we're going to separate the sister chromatids from one another. So in other words, this is going to be one cell, that's going to be one cell, that's going to be another cell, and that's going to be the 4th cell. So what you see, these little 4 individual unique pieces of DNA, that's what eventually is going to make up the gametes. And so, yeah, Daniel.
I'm sorry.
It's okay.
That all happens in...
Sorry, that all happens in the very first stage of meiosis 1. That happens in prophase 1.
Crossing over. So to clarify, crossing over will be one chromatid of each chromosome.
Switching pieces of DNA with one another.
And then the chromosome is split in half.
Yeah, and then the rest of meiosis 1 is pretty similar to what happens in mitosis. So the same things that happen in mitosis are going to happen in meiosis. So what's the first thing that happens in mitosis? Prophase, in which the DNA condenses, right? So the DNA is still going to condense in prophase of meiosis.
This is the crossing over already.
It's the crossing over.
Already has happened, and then you can go ahead and finish the cell cycle.
Yeah, the only thing that's really significantly different in meiosis is that there's 2 cell divisions, and then there's this crossing over. And that crossing over is extremely important because it adds to the genetic diversity of individuals. And later on, I'll give you all a stat about actually how unique and individual you are. But, so in prophase 1, so think of the cell cycle, we've gone through G1, we've replicated our DNA, we're still making, sister chromatids, go through G2, go through all the checkpoints, so now we're ready to enter into meiosis. So the first thing that's going to happen, prophase 1. In prophase 1, the DNA is going to condense, and this crossing over is going to occur. And it's kind of shown in this image right here. It's not very blown up. I think there's a slide that has a bigger version of it that we'll talk about. But anyway, so the DNA is going to start to condense. You're going to get this crossing over. The nuclear envelope is starting to degrade. And then in metaphase, instead of just the individual chromosomes lining up on the metaphase in the middle of the cell, now we're lining up the homologous chromosomes. So here's a pair of homologous chromosomes. This one from mom, this one's from dad. And then here's another pair that have crossed over. This one mostly mom and this one mostly dad. And so during anaphase 1, you're pulling those those homologous chromosomes into opposite sides of the cell. And then in telophase, again, you start to get this pinching. You start to remake the nucleus. I'm sorry, what?
The furrow.
The furrow. Yeah, this is an animal cell, so it's got the furrow right there. But anyways, we're dividing the homologous chromosomes from one another. And so at this point, right here, these are diploid. I'm sorry, they are not diploid, they are haploid. They still have 2 chromosomes in them, but they're different chromosomes, right? If you had chromosome 1 and chromosome 1 from mom and chromosome 1 from dad in the same cell, it would be haploid. But since we only have one or the other, that's why it's going to be diploid. Y'all see the difference between the 2?
I believe in now haploid.
Yes, it's haploid after the first
You just said
meiosis.
they would die.
Did I say that? Sorry. I can't talk today. All right, after meiosis 1, they're haploid. And they stay haploid after
Boy.
meiosis 2. So they start off diploid, and they become haploid, and then they stay haploid after that. And so a lot of the major, functions of these stages, like anaphase, the exact same thing. You still have the spindle apparatus, you still got the centrosomes and the microtubules and attaching and pulling, and all that's the same. It's just what you're pulling apart and the crossing over that's different. And so after you go through meiosis 1, you're going to end up with 2 cells, and they're going to have half the amount of genetic information in them. So now it'll go into meiosis 2. And by the way, there's not another DNA replication, because in the cell cycle, you still got your G1, your S, your G2, and your M, but your M is made up of metaphase 1 and metaphase 2. So you're not going back around, you're still staying in the M phase when you go from meiosis 1 to meiosis 2. So you're not going to replicate your DNA again. And so in meiosis 2, again, here are the 2 cells that we started with. Each one are going to be haploid. Same thing, prophase, DNA is going to condense. You're going to start losing the nuclear envelope. Metaphase, all the chromosomes are going to line up in the middle of the cell. Anaphase, you're separating the sister chromatids just like you do in mitosis. And then in telophase, you start to get the pinching, the cleavage furrow, and the new formation of the nucleus. And then eventually you end up with 4 cells. But remember, we started with these homologous pieces of DNA. And so after 2 rounds of division, we're going to end up with 4 different cells. So let's just say we start with a cell that only has 1 pair of chromosomes. So that's our cell. We're we don't have a lot of DNA. This is not an intelligent organism. It just has 1 chromosome, 1 set of chromosomes. But anyways, after crossing over occurs and you go through meiosis 1 and you go through meiosis 2, now you're going to have 2 different pieces of DNA. So this is going to be 1, this chromatid right there. This guy is going to be another, this chromatid right here, number 2. This guy's number 3, and this guy's number 4. So our other one, and then finally this guy. So you can see how they're all individual, genetically different from one another. And so each time we go through meiosis, we make 4 different versions of an egg or a sperm. So this is 1 individual sperm, this is another individual sperm. Just like this is 1 egg, this is a totally different egg. And so, and based on the way that the homologous chromosomes are aligned in metaphase 1, so going back to meiosis 1, so you notice here that in this case we have dad's, number 2 on top and mom's on bottom, whereas here we have mom's on top and dad's on bottom. So the way that these orient themselves is going to be totally random. So you could potentially end up with a cell that had all 23 of dad's chromosomes in the bottom and all 23 of mom's on the top. Or you could have half and half or any other spectrum. Basically, since there's only 2 ways that each pair of chromosomes can be arranged, and we have 23 different chromosomes, so if you do the math, that means there's this many different combinations of chromosomes for a human cell based on how they line up with respect to other homologous chromosomes. This number, it's like 8.2 billion.
Different variations.
Different possibilities, probabilities. So every time you make a sperm, you're going to get something that's 1 out of 8.2 million. Or an egg that's 1 out of 8.2 million. And so that means in fertilization,
Why, that's a
Is it
million.
million or billion?
I said,
You
It
said
is-
million. You wrote billion.
I tend to do that. I believe it's 8 million. It's 8 million. It's 8 million. Sorry. I'm confusing this with my length of DNA stat. So 8.2 million. So that's one egg or one sperm. So think about fertilization. When you have one sperm, that's 1 out of 8 million. And one egg, that's 1 out of 8 million. And you make an individual, whatever 8.2 million times 8.2 million is, a crap ton of million. Then that's how genetically unique you are. So you literally are like 1 in a million.
That's 1 out of 67.24 million.
1 out of 67.24 million. That's
So-
your genetics right
So
there.
there's likely to be at least one more of you than there.
Possibly, but they would have different parents, right? So if a couple, if they had, what was it, 67 whatever,
67.24 million.
yeah. If they had that many kids, 2 of them would be the same.
There isn't that many months in a
I know,
lifetime.
I'm just saying, theoretically. So anyways, we're all super genetically unique, which is why we all have the same genes, but we all look and act and behave so differently from one another.
It's completely intense.
All right. No, that's not going to be on the test. It's just a fun fact.
I like fun facts.
Let's have some more fun facts.
More fun facts? I'll see what I can come up with. Okay, so just to go through the steps again, in the first division in meiosis, meiosis 1, you're going to start with prophase 1. And in prophase, what always happens in prophase? The DNA always condenses, right? And then in meiosis, we're going to have that crossing over event between the homologous chromosomes. Other than that, prophase is the same. Metaphase is going to be the same. You're still going to have these chromosomes lined up along the middle, but instead of having all the chromosomes stacked on top of one another, you're going to have all the homologs right next to one another. And so the mitotic spindle is still being set up, or it's actually complete in metaphase 1. And then in anaphase 1, this is when you pull apart the homologous chromosomes. And then that's going to be followed by telophase, no cleavage furrow, and then you end up with 2 cells. And those 2 cells are going to be haploid, have 1 set of genetic information. And then in meiosis 2 is going to look exactly like mitosis. So in meiosis 2, instead of separating the homologous chromosomes, you already did that in meiosis 1. So in meiosis 2, now what you're doing is you're splitting those sister chromatids from one another. And remember, you've already gone through crossing over, so those sister chromatids are more than likely going to be 2 different versions of one another, which is how we end up getting 4 different cells, all that are genetically different. Yeah, Alan?
Real quick, you said that it never goes back to the interface stage, G1S and then G2. So what happens is, I guess after meiosis 1, if I'm correct, it just goes right back after the M stage, right back into 2.
Goes right back into 2. So it's just, think of it as almost like 2 individual mitoses. You know, the first one separates homologous chromosomes and has this crossing over, and then it divides and, telophase, and then you have cells. And then they start immediately condensing again. They took all this energy to reform the nuclear envelope in telophase 1, and then they're just going to turn around and break it back down again in prophase 2, or condense it down, and then, so it's literally one right after the other. Yeah?
Okay, you said that the chromosomes or chromatids condense
Yeah.
in prophase, right?
Yes.
On the test, the answer was anaphase.
Does not condense and,
It says at what stage is mitosis, in what stage of mitosis does chromatin condense to become chromosome?
and I put the answer key says,
The answer key says D. And
which is not,
the answer is prometaphase.
Oh.
Phase, and it's the anaphase is procancer.
really? Okay, what number is it?
17.
Oh, that's the one we had issues with, yeah. Okay, well, I'll go back and I'll make test corrections, which means you need to turn your test back into me, so I can go back and add points to it.
So if we got it right, you want it wrong?
Yeah, if you got it right, no, don't mark it wrong. Don't mark it wrong if you got it right.
What
I got it
number?
right to your.
The correct answer is definitely prophase.
For what?
Is it pro-metaphase
17?
or
Well,
prophase?
It's pro-metaphase.
Prometaphase is where you can see the chromosomes.
But we had omitted prometaphase.
It's, like I said, the stuff that's occurring, it's still occurring. So it's not like
Pro-metaphase?
the steps involved in prometaphase just disappeared. It's just the title prometaphase disappeared. That's the only thing that changes. So anyways, after class, turn in your test. If you got it wrong, it should be right. Turn them in. I'll make the corrections.
What should it be?
So if we put pro-metaphase, it's wrong.
If, I think I changed it, so the options are what?
Pro-metaphase, prophase.
Metaphase and anaphase.
I thought I took out prophase. I didn't.
Yeah, you said when it became visible.
Yeah, so the answer should be prometaphase, because that's talking about
And that's
mitosis,
what
right?
three of us put.
Yeah, I know, so I
Does the chromatin
I must have
I
made a
still want to
mistake.
turn it in if I
become
had-
chromosomes?
What?
It says at what stage does chromatin condense to become chromosomes?
Yeah, so prometaphase.
The answer you put on the scan card.
I'm aware of that,
Anaphase,
which is
right?
why you're gonna turn it in, and I'm gonna correct it and give you 2 more points, if you got it counted wrong.
So it has to be prometaphase.
If it's wrong, turn
What
it.
about if you put
If
metaphase?
it's wrong, turn it in.
I said pro-metaphase, and that's right.
Well then,
So if it's prophase, it's not right.
Then you
Just keep
Because-
repeating.
broke it.
17 should be D. That's what I have.
Because it was letters were D, E.
But these.
but I told y'all to make it A, B, C, D.
So if you didn't make it A, B, C, D, you got it right. So we should have all left it as D,
Actually,
C, D, C.
prometaphase would be. Prometaphase should be A.
That's why I give you curves for questions like this. They're kind of like, in retrospect, not.
Clear curve.
So,
All right, we can talk about it after.
Everyone
Let's
plus
get back
2
to this. Okay,
3 points for everybody.
So you all circled A.
I circled C. That's wrong either way, right?
anyways, so, in prophase 1, again, this is where this crossing over is occurring. And so this is a diagram used to kind of illustrate that. So like we said, homologous chromosomes, they have the same genes. So they'll both have gene A, gene B, gene C, gene D, gene E, so forth and so on. And because they're so similar to DNA sequence, you're able to just kind of switch places with one another. You just kind of cut and paste different pieces of the chromosome together. So this is the crossing over. This is where you get these non-sister chromatids crossing over with one another. And again, they end up swapping genetic information. And so remember, in meiosis 1, we're separating the homologous chromosome. So this will be in one cell, and this will be in a different cell. And then meiosis 2, we're separating the sister chromatids. So that's how we end up, this will be in one cell, and then this chromatid will be in a different cell. Just like this chromatid will be in one cell, and the other one will be in a totally different one. So that's how we end up with 4 genetically unique gametes. And so those pieces that we cut and paste, the chromatids that did the crossing over, we call them recombinants because their DNA's been recombined between 2 different sources. And the other 2, the ones that look exactly like the original parents, those are called the parentals. So for every round of meiosis, you're going to end up with 2 parentals, which are going to look like the original parents. And then you're going to have 2 recombinants. Those are the pieces that went through the crossing over in prophase 1. Okay,
No.
no video?
No,
Yes, you just
I just
wouldn't
wanted
have-
you to go back.
Okay.
So is it technically possible?
Oh, I forgot y'all don't have the slide, I'm sorry. Is it
No,
technically
no.
possible what?
We do have the slides, they're online.
What? Yeah, you have them, you didn't have them printed out though.
Never mind. It was a stupid question that I know the right answer to.
Am I good? Yes, okay. Was that a yes?
Yes.
All right.
Crossing over during meiosis allows recombination of genes between homologous chromosomes. This alters the linkage between genes on the same chromosome. Crossing over occurs frequently during prophase 1 of meiosis, when homologous chromosomes associate with each other in a structure called a tetrad. First, consider the possible inheritance of alleles for 2 genes on a particular chromosome when crossing over does not occur. Recall that one member of each pair of homologous chromosomes came from one parent. The other member came from the other parent. In this example, one parental chromosome had alleles capital A and capital B, while the other had alleles lowercase a and lowercase b. Without crossing over, each haploid cell produced will inherit a chromosome with the same combination of alleles as was on one of the parental chromosomes. Thus, the arrangement of linked genes is not altered in this case. Now, consider the case where there is crossing over in the region between the 2 genes. Since the crossing over is only between 2 individual chromatids, the possible haploid cells after meiosis have 4 different genetic combinations. 2 will come from the chromatids that did not participate in crossing over and thus will still have the parental combination of alleles, either capital A and capital B, or lowercase a and lowercase b. The other 2 will be recombinant with either capital A and lowercase b, or lowercase a and capital B alleles. Thus, crossing over allows for the reassortment of linked genes.
All right, did that make any sense at all? Maybe.
Kind of.
Kind of?
So can you ever have it so that,
Crossing over happens in prophase 1.
can it ever happen that it happens from all of the chromosomes? Like they all cross over?
It's random. Sometimes they'll cross over, sometimes they won't. Sometimes they'll cross over 2 times. Sometimes only one tail will cross over. Other times